Okay, well, I guess we'll get started then. I'd like to thank you all for showing up this evening. I'm going to ask people just to mute for the moment if you're not speaking. So this is a very basic introduction to the workshop at Hackathon that we're going to be holding in a few weeks on multi-scale virtual tissue modeling using CompuCell3D. The purpose of this workshop is to give you a little bit of a taste for what we're going to be doing, help people get an idea of material that they might want to work on to prepare for that workshop, and also just to get to know each other a little bit and to familiarize you with some of the resources that we have. I should tell you that all of the workshop materials are going to be live streamed on YouTube and recorded and distributed. So please be aware that everything that we do in these workshops is public. I'd like to also thank our funding sources, the National Institutes of Health and the National Science Foundation, and Nanohub for their contributions over the years. And one thing that one definitely wants to do by the end of today, perhaps, or if you're in a bad time zone tomorrow, is just to make sure that you could either run Compusell online through Nanohub or else download it and have it run on your local computer. That's something that the sooner one has that taken care of, the less of an anxiety it is as we go forward. During this little presentation, if you have questions, please put them in the chat and we'll do our best to answer them. And then we'll have time, if people are still awake, given where you're coming from, we'll have time for some live questions as well. So I'm going to give you a little bit of course logistics, a little bit on background knowledge and reading, and then we're going to introduce very quickly Compusell and how it works. We'll build a quick simulation just to see what it feels like and talk about how simulation specifications work. And we'll talk a little bit about the hackathon. We have officially about 130 people registered so far. Many of those people for a free online course. Many people who register don't show up. So it's certainly possible that the total number of people is fewer than that. But we will do our best to support each of you. But if we really have 130 people who show up, it may take some time to get around with our staff to answer every question. So we'll do our best. So please be patient. If it happens that you're an experienced Compusell user, because we do get some people who have Compusell experience who take this course, and you're willing to help mentor beginners, we definitely could use your help. And if so, please let Hayden Fennell here, hfennell@iu.edu, know that you would be interested in doing that. So I'd like to welcome you and remind you that the theme of this course is on multicellular modeling, and the multicellular modeling has all sorts of biological applications. This methodology could also be applied in material science, and but it's primarily applied in computational biology, asking questions about how tissues are organized, either during development, the various stages going from an egg to an adult organism, homeostasis, how does an organism maintain itself? Somebody pointed out the other day that this was a picture of a mouse among the same picture of a mouse. That's not trivial in the life cycle of an organism. How do you maintain an organism? Developmental diseases like cancer, which involve failures of developmental and maintenance mechanisms. Infectious diseases, where you have a pathogen that enters the organism, spreads, leads to an immune response, and causes both direct and indirect tissue damage and needs recovery afterwards. Toxicology, how do you maintain or build an organism in the face of chemical insults in the environment? And then in medicine, how do we design therapies using the insights that we gain from these models? And potentially, how can we use this kind of modeling in context of bioengineering, either to build replica biological systems or potentially to develop totally new of biological constructs. When we come to dealing with biological modeling, we have to face the fact that we have a range of scales that's intractable. We can't build a computational model of every cell in an organism or every molecule in a cell. And therefore, we're going to have to pick subsets of scales that we're going to work on. Typically, in the world that we live in, we're going to start with cells as active agents because these are agent-based models. We're going to ask, how are those cells controlled at the molecular level? What are the networks that control those cells and their behaviors? How do those cells interact with each other and with the outside environment? And how does that all play out in the context of tissue organization and function? And so that range of scales from subcellular up to tissue is the focus of the kinds of models that we're going to be developing. That could be timescales in our world from a micron to centimeters, and timescales on the order typically of minutes to years. We're not going to be worrying about molecular scales. We're not going to be worrying about molecular dynamics simulations, and we're not typically going to be doing whole body biomechanics, although some of those are accessible at either end. As an example here, which I always show in this demonstration, this is an electron micrograph of a chicken embryo. This shows the process of seminogenesis or segmentation when the periodic structure of the vertebrae is going to be laid down. We have an understanding, we think, of some of the signaling and gene regulatory networks that are operating inside the individual cells in this tissue. Those are oscillatory gene regulatory networks. And we can see in the simulation here on the right, we're monitoring in the simulation the level of one particular oscillating gene. You can see the pulses of the gene expression here. That gene expression interacts with a gradient of a chemical signal, a morphogen, in this case, a growth factor, fibroblast growth factor, FGF8, which is high in the posterior of the animal, low in the anterior of the animal. There is a differentiation process, which is determined by the level of FGF8 and of the oscillating genes in the cell. That leads to a change of cell motility and cell adhesion that leads to rearrangement of the tissue to form these quasi-spherical organizations. And that would be a typical developmental biology application of this kind of methodology. These kinds of methods can be used, though, much more widely. Here's an example. All of these are CompuCell3D simulations. A CompuCell example trying to understand formation of kidney tubules and their stability. Polycystic kidney disease is a proliferative disease in which the tubules have inappropriate cell growth, leading to a cystic phenotype and eventually leading to death due to kidney failure. It's a relatively rare genetic disease, but it's lethal for people who have it unless they have kidney transplant. Here we have a more classic abstract model of a tumor. The tumor is growing here in the context of vasculature. The vasculature is supplying oxygen and glucose to the tumor. The tumor develops a hypoxic core, which sends out a growth factor, a vascular growth factor that induces neovascularization in the tumor environment, which then supplies additional nutrients and oxygen to the tumor. This is a more biomedical application. We have a patient-derived image of blood vessels in the retina. We calculate the oxygen tension in the retina as a function of position. And in diabetic retinopathy, we have loss of blood vessels due to damage from the diabetes. That loss of blood vessels can lead to cascading failures of vasculature, leading to blindness. It's treated by laser ablation, and here is a simulation trying to address what pattern of laser ablation does the minimal damage to the intact tissue while maximally preventing the spread of the damage. Here on the right is another vascular simulation, this time in 3D. This is a case not of loss of blood vessels on the left, but rather of inappropriate growth of blood vessels. In this case, blood vessels from behind the retina cross the membrane that normally separates the blood vessels from the retina, called Brooks membrane. Once they get into the retina, they begin to proliferate and invade, and they'll eventually cause the breakdown of the layered structure of the retina and eventually, again, blindness. This is a disease that's pretty common in the elderly. Diabetic retinopathy and retinal neovascularization subsequent to age-related macular degeneration are two of the main causes of blindness in the developed world. In poorer parts of the world, there are parasitic and other kinds of diseases that cause a lot of adult blindness as well. But in the United States or Australia or New Zealand, these would be the two main causes of adult blindness. In both cases, the mechanisms were not clear when we began this study, so trying to understand how these diseases work. This kind of methodology is also used quite a bit by the Environmental Protection Agency here in the United States, trying to understand toxicity mechanisms, in particular toxicity during development. There's some nice papers using Compusell, trying to understand disruption of blood vessels. You'll see that blood vessels are a typical theme in CompuCell simulations, there are a lot of them. Here of the development of the urogenital tract, particular estrogen mimics can lead to feminization of males during development. And this is a study of how that works or fails to work by Max Long. It's a nice work that he did. And this is another study also of developmental defects by Shane Hudson at Vanderbilt, who's probably going to come and speak about this work to the workshop, looking at palate fusion. So in the developing embryo in humans, also in mice, other mammals, the roof of the mouth is formed from two bones that initially come from the sides, grow in and fuse together along the midline, which is why you have a ridge at the top of your mouth. That growth and fusion is controlled by a variety of different cell signaling molecules. And this is a simulation of disruption to that development by toxicants in the environment that can lead to cleft palate syndrome in children. So this is an example of the toxicological developmental aspects of this kind of simulation. Another thing that people have worked on quite a bit in the past couple of years, mainly post-COVID, although people use these modeling methods also pre-COVID, would be to look, for example, at a patch of lung tissue here in blue, where you have an initial infected cell. Here we imagine infection by a virus. The virus spreads in the tissue. The cells die from the infection. They also call in immune cells. Those immune cells, in turn, kill both infected and uninfected cells. And you can ask questions about how things like antivirals or immune modulators affect the balance between preventing the infection from spreading and the tissue damage caused by the immune response itself. As we know, if you have a disease like influenza, the symptoms that you experience of influenza are actually not caused by the virus itself. They're caused by our immune response to the virus. Of course, if we didn't have the immune response to the virus, we'd die from the virus. But the balance there is quite complicated. And this is the kind of methodology one can use to investigate those kinds of questions. By the end of the course, we hope you'll be able to do three things. The first is to look at a biological problem We've talked about a variety of biological problems, and begin to ask quantitative questions that you can use simulations to answer. Simulations are not magic. They're able to do certain things very well, but they also have limitations, and they're a lot of work to develop. And so it's important that we understand what kinds of questions are appropriate to answer with these kinds of methodologies, what kinds are better left to pure experimental methods, or other kinds of computational methodologies like bioinformatics. Once you've identified a problem, we'll talk about how to analyze that problem into the components of the system. Multicellular systems, we have to identify what the players are: cells, cell types, extracellular matrix, diffusible chemicals, how they interact. And eventually we have to be able to formalize that into mathematical models. And then we'll, on the technical side, talk about how to use CompuCell tools to build computer simulations that implement these models that we've designed, and a little bit about how to display and analyze simulation results to give biologically meaningful outputs. So those are really, there are many other things that we hope to learn too, but those fundamentally are the goals, educational outcome goals of this workshop. A little bit about course logistics. There's going to be a Zoom link. We'll use the same Zoom link as today for the workshop and the hackathon. There may be some additional Zoom links during the workshop for 1 or 2 days. If that's the case, we'll provide the Zoom links, and we'll remind people of these links. I hope people were able to get in. If you're here, you must have been able to get in, but I hope it wasn't difficult to register for the Zoom. There is going to be a shared document for people with hackathon ideas to work on those. And there's also a Slack channel that I hope will help people build a bit of community and ask questions and answer questions, even without being face-to-face. And the Slack is particularly valuable for people who are in awkward time zones because you can ask questions and have them answered offline. I think everybody here is probably familiar with Zoom. We've all used it enough in the past few years. Please feel free to use the yes, no, go slower, go faster, raise hand buttons in Zoom. I'm going to be doing a lot of polling, which will be asking, are you doing the exercise? How is it going? Do you need more time? Do you need help? All the exercises in the class will be available in Google Doc format, so you can work on them offline. All the slide decks will be available for download, and the videos will be made available as soon as possible. There'll be raw versions up right away, and then there'll be edited versions in the next day or two after each session. Again, all sessions will be live streamed on YouTube and recorded. You can see previous year's courses if you like, 2021, 2020. Some of the material will be the same, some will be different. So those are always available to you as resources. We have a large number of volunteers who work on this course. I'm James Glazier, Professor of Intelligent Systems Engineering, head of the Biocomplexity Institute here at Indiana University. TJ Seago is the lead developer of Compusell, a great person to go to if there are technical problems with the software. Professor Gilberto Tomas and Pedro Del Castel from Brazil are big CompuCell users and great instructors. They'll be helping teach the course, helping people out. Julio Belmonte from North Carolina State University will be giving a bunch of lectures, another major CompuCell user. Jim Sluka here at IU is a specialist in toxicology, also bioinformatics, can help interface between different disciplines. Joshua Aponte Serrano and Priyam Adyapok are both postdocs who've used CompuCell a lot. Josh is now at the NIH. Priyam is at Duke. And Hayden Fennell, who's here at the top left of our screen, is an expert in education using computer simulations. He's been helping us out on all aspects of the course. There'll be other people as well who'll be helping out, and their names will be added to the roster as the course is finalized. The daily course schedule, and again, I apologize to our collaborators and friends in Australia and New Zealand and India, is going to be the following. We're going to have a brief opportunity for questions, informal troubleshooting before each day. Each day will consist of 4 90-minute modules, starting at 10 a.m. in Eastern Daylight Time, UTC -4. Each module will typically consist of 30 to 45 minutes of lecture with 45 to 60 minutes of hands-on exercises working together. We may have some particular modules that have more computation and exercises, some more lecture, but that's the desired balance. There'll be a fairly long midday break, part of which we will use for extra material, troubleshooting, hackathon organization, a lunch break, and then 2 afternoon modules, and then afterwards an opportunity for people to ask questions and do more troubleshooting online. My experience, by the time 6 p.m. rolls around, people are tired after that many hours of work, but we're always available. The instructors are always available to work with you and support you. In terms of subjects, we begin with a day 0 on Sunday, the July 31st, which will be a Python tutorial refresher. Many of you may be Python experts. If you are only a Python moderate or beginner, or you haven't used Python before, we strongly recommend that you refresh your understanding with that course. Monday, we will start by talking about Compusell, some repetition from today, and beginning to explain how Compusell works internally and using it to build cell models. The second day, we begin to talk about chemical fields and the concepts of modeling. The third day, we couple chemical fields to cells, both for movement and for growth and control, build an avascular tumor simulation. We'll start thinking about subcellular regulation networks, signaling and gene regulatory networks, and how to implement them and put them into CompuCell cells. Day 4, we'll continue that and start talking about structure with inside of cells. So we'll talk about how to model, say, the nucleus of a cell, how to model junctional adhesions and filopodia, how to model epithelial cells and polarization. And then in day 5, we'll continue with these merging of topics and also talking about the use of CompuCell to do large-scale simulations on clusters with CompuCell Python optimization and parameter fitting. You'll notice we have module 3 on both days 4 and 5. Those are going to be presentations by people who have used CompuCell to do advanced modeling and published these results. And they're going to be presenting the science of what they do and also walking you through their code to show you how to build more sophisticated models. So we're going to be doing basic CompuCell in the main lectures with examples of more sophisticated models included. And then days 6 and 7 will be a 2-day hackathon. To allow you to try building a model and developing it as you like. We recommend that you do some preparation before the workshop starts. It's not required, but in our experience, while we try to cover everything within the workshop's week, things move pretty fast. And the more preparation you have, the more likely it is that you have a good experience with the workshop. We'll do our best to be available both before the workshop, during the workshop, and after the workshop to help with scientific and technical questions. When you think about the kinds of projects that we're doing, they involve a lot of different things. You need to know some principles of modeling. You need to know some Python programming because we use Python scripting. You definitely need to know some biology. Knowing some biophysics is helpful, although not essential, and knowing some mathematics is helpful. You don't need to know any of these topics in depth, but you do need to know something about all of them. And typically we find that nobody comes to this course unless they've been doing this kind of modeling already in detail. Nobody comes to this course with all of the background that they need to build and use these kinds of models. The audience for this course is highly diverse. Over the years, we've had everything from high school students to emeritus professors take the course. We've had everything from pure mathematicians to surgeons take the course. And that diversity is a challenge because we try to come up with material that's accessible to everybody. It's also a great strength because it means people can work together and complement each other. And in the days when we did this in person, we did teaming throughout the course to have people help each other. The hackathon is partly a successor to that, recognizing that teams composed of people with biological wet bench experience, for example, or medical experience, can work very productively with people with more mathematical experience or physical experience or computational experience to learn from each other and help build the understanding needed to build these complex models. Talking about principles of modeling, the key thing is to start by trying to ask what kinds of questions can be answered by models. Then we have to be able to understand how to quantify those things. What are meaningful measurements that we could do in an experiment or in a simulation that could be compared to each other? And then we have to do something that biologists do naturally, which is build abstractions, build models. And wet lab biologists and clinicians often say they don't use models, but in fact, modeling is intrinsic to biological practice. Perhaps not computational and mathematical modeling, but anytime you have a beautiful drawing of a biological system, you have what's called a conceptual model. And so here in this example, I have a picture of a liver, and then there is a schematic showing the flow in the liver. And I could use this schematic to ask the question, if I have a chemical in my blood, how are different parts of the liver exposed to different concentrations of that chemical? That's pretty important if I'm doing drug dosing, for example, or drug elimination studies. And then here I have a mathematical model embodying that flow in a simple way. And so this process of going from biological observation and reality to a conceptual abstraction, to a mathematical abstraction, is something that's challenging and is something that I hope everybody will learn during the workshop. Deciding which components are important, what level of detail to represent them at is more of an art than a science, something that we have to build intuition about. Thinking about how to map real-world objects and mechanisms to mathematical abstractions, that's the second phase here, is also sometimes challenging. And when we're going to be building subcellular models, we have to be able to think about how to build network diagrams and what they mean. We want to be able to think a little bit about the limitations of these models because they're always very great simplifications of reality, how to validate or falsify models, and how to use them to explore mechanistic hypotheses. One of the things that we'll see repeatedly in the course is a diagram like this, talking about how models are built. All models are constructed of a list of the objects that are being modeled, the properties that are related to those objects or their states, those objects' intrinsic behaviors and interactions, how those things change in time, the dynamics, that is continuous time events, and events in discrete time. An example of a dynamic process would be the level of ATP change inside of a cell. An example of an event would be a cell death or a cell division. And then we have to think, because we're not simulating the entire history of the universe or the entire universe, we have to think about where our modeled simulation bounds to the outside world, either in time or in space. What are the initial conditions of our simulation? Where are the elements? What are their states? What happens at the edges of our simulation? And if we're modeling something like drug treatment, what's the pattern of drug treatment and how is that introduced? Something like radiation therapy model would also have complex temporal conditions as well. As I mentioned, most of CompuCell models use Python scripting. If you've used another language like MATLAB, C++, Mathematica, Python won't be that different. The basic syntax is pretty straightforward. The one thing that you have to get used to with Python is that indentation is used instead of parentheses to define blocks of code. It's easy to get lost with indentation. You need to know about how loops work. Those are pretty basic, but they're a little bit different in Python from most other languages because you loop over lists rather than over sequences. You need to know about things like ifs and conditionals, object types. And there are two particular types of object that are very important in CompuCell: lists and dictionaries. Lists are pretty general computational concept. Dictionaries are a little bit more specific. CompuCell uses dictionaries as a way of communicating information between cell agents and between different layers of the simulation. And those are really the core Python concepts. Everything else that's fancy Python, you don't use much. In CompuCell simulations, CompuCell is designed to try to hide as much of the programming complexity as possible from the user. Again, while the level of Python that you're going to need is not too much, it is important that you be comfortable with the basics. And so we recommend that if you haven't used Python before, you do a little bit of preparatory work or take the Python bootcamp or both, just to make sure that you don't get stuck on those elements of the simulation. Biology, of course, is the core of what we do. We're talking particularly about cell biology and tissue biology, so understanding what cells are and what they do is pretty important. Again, we're talking about Wikipedia level understanding of processes, but how do cells generate forces? What determines when they divide? What determines when they die? How are tissues constructed by the interactions of cells and their environment? What are some basic concepts like mesenchymal and epithelial cells, mesenchymal and epithelial tissues? Concepts of things like cell polarity and planar polarity. These are some basic biological concepts that we'll use repeatedly during the course. They'll be very familiar if you come from a biological background, but for people coming from mathematics, physics, or computing, maybe not as familiar. What is extracellular matrix and how does it work? What are these signaling, metabolic, and regulatory networks operating inside of cells? And how do those networks then control cell behaviors, which is still a bit of an open scientific problem in many cases. There are some basic questions about networks, like network motifs, that are pretty important, that we won't have time to go into great detail in this course. And then how do we characterize at the cell level things like blood vessels, vasculature, which you've heard talked about already, and other systems properties like cancer behaviors. And then something that we've already alluded to, which is how do biologists represent information? And of course, if you're a bioinformatician, you think about large data sets and filtering. But classically, biologists represent information in the form of diagrams. And one of the things about CompuCell, as you've seen from the examples that I've shown before, is that it's a very visual way of interacting with the world. It's helpful, although not absolutely essential, to have some basic physics understanding, biophysics understanding. In particular, CompuCell is built around the idea of constraints. And so understanding what a constraint is and how they work, and you could think of a constraint as a spring, for example, which constrains 2 points to be at a given distance from each other, or even a balloon filled with gas, where if I squeeze on the balloon, it becomes smaller. If I pull on the balloon, it becomes bigger. Those are simple examples of constraints in physics. A key concept that we'll need is diffusion. Chemicals diffuse in the extracellular environment within cells, particularly what a diffusion length and a diffusion time are important concepts. How forces and velocities relate to each other is quite helpful. The motor under the hood in Compusell is something called the cellular POTS or GGH model. This is a statistical mechanics model. You don't need to know how that works to use Compusell, but when Compusell misbehaves, knowing a little bit about the internals help you diagnose the problem and fix it. So it's a little bit like driving a car. When the car is working, you don't need to know how the engine works. But when the engine fails, if we're going to fix it, it's helpful to know what a spark plug is. A lot of these functions are stochastic. Biology is stochastic, has a lot of randomness in it. And knowing a little bit about how randomness works in physics is quite helpful. We try to hide as much mathematics as possible. It is useful to know what first order ordinary differential equations are. dx by dt equals something. That's really helpful. The concept of a rate law, how fast something occurs and how it depends on parameters, is helpful. Partial differential equations, primarily diffusion equation, which we saw in the last slide, very useful. How to translate stochastic probabilistic transitions into rates back and forth is helpful. And when we're dealing with subcellular signals, understanding chemical reactions and their representation, stability and fixed points and sensitivity is helpful. We will not do a lot of that in this course, but if one is interested, Herbert Sauro from the University of Washington teaches a companion course to this in the third week of July on network modeling. We often, in the old days when this was done face-to-face, would team teach it here in Bloomington, Indiana, as a 2-week course or sometimes an extended 1-week course. And I encourage you to look at Herbert's course if you're interested in learning that material. Again, almost nobody comes to this material knowing everything to begin with. We have some material that we recommend, depending on where you feel you need to fill in. There's this nice paper on principles of modeling. It was written by some Canadian graduate students a year or two ago. Rather different context and some different ideas, but still very helpful if people have not done mathematical modeling before. We have a draft paper of ours that outlines some more specific considerations for building models in the context of multicellular models. There are a number of good Python online textbooks. If you want to find open source free exercises for Python, we think the Rosalind program is really excellent. In particular, their first 6 exercises, Python Village, are a great way to prep for the course. Network modeling, tellurium and antimony are the languages we use. There's a great tutorial available online, tellurium readthedocs.io, quickstart.html. You could take Herbert's course too, if you have a week extra to do that. And he's also written a great textbook, Systems Biology Introduction to Pathway Modeling, which I strongly recommend. There are many good places to learn the basic mathematics. This is a textbook that we find is good for people not too demanding, but Wikipedia actually has a lot of the basics too. On biology, I really love Gilbert's developmental biology textbook. It really covers an awful lot of the things that we need to know at about the right level. And if you don't own a copy, I recommend buying an old copy. You can go online to Amazon.com or another secondhand bookstore and you can hold on a second. And you can buy for $2 or $3 a secondhand copy. The principles of developmental biology have not changed, but the level of molecular detail in the newer editions is confusing. And so if you're a beginner, I actually find the older editions much more easy to read. For physics, there's a relatively elementary textbook. Phil Nelson's Physical Models of Living Systems is not a bad entree into this field. If you really want to dig in, Phillips' Physical Biology of the Cell is a fantastic textbook, but it is relatively demanding. So if you have a strong physics background, I strongly recommend that, Physical Biology of the Cell. We don't yet have a textbook on our modeling methodologies. We should probably be writing one. But we have a paper, which I'll give another link to in a little bit minute, which gives you the under the hood details, and we recommend that. If you find any material that you think is useful and that you think your collaborators and colleagues would find useful too, please send them our way, and we will make sure that that information is distributed to everybody in the course. Resources. Again, there's the CompuCell website. We'll try to keep that updated with current material. There are the online manuals, read the docs, which are helpful. If you want to try doing some exercises on your own, there's the CCD3D Quick Start Guide. This is an old document. It hasn't been updated since 2011, but the basic concepts haven't changed, and you can still do the exercises. They still work. We have our Slack channel for people to work with. Google Drive will have all the course materials in it, exercises, lectures, and so on. And then there for Tellurium, for network modeling, that read the docs that I mentioned. There also are a series of quick reference guides that we'll come back to in just a second. There'll be a Slack channel for people to communicate with the instructors and with each other. Please join that as soon as possible to get set up. And we strongly encourage people to use that, and it's be available after the course ends too for people who want to work on projects and ask questions. The quick reference guides, there are 3 of them. One for basic Python programming, one for CompuCell3D, and one for Tellurium antimony modeling of networks. I strongly recommend that everybody print them out and have hard copies of those 3 on their desktops while they are taking the course. It's challenging to switch back and forth between the simulation environment you're working in, the lectures that are being given online, and the manuals. And so having the manuals printed out is very helpful. We'll try to teach the course in a way that works for people with a single screen or a laptop. But if you have the luxury of having 2 screens, a secondary screen where you can have the PowerPoint or Zooms on one screen and your computation on another screen, that will make the course easier on you and probably easier on your eyes too. I mentioned that we have an article, a fairly old article that talks about the mechanics of how CompuCell works under the hood. That's available to you. And I do recommend that nice paper by those Canadian grad students on how to build mathematical models. Again, they're thinking about other kinds of models, not the ones we're going to be developing, but the principles are quite useful. We're going to have a hackathon. I don't know, maybe we could have a show of hands using the response button about how many people were thinking about participating in the hackathon. We're hoping that people, at least one person is saying yes. We're hoping that we're hoping that people will come up with some good ideas and lead some teams. You don't have to have your own idea to join a team. You don't have to have any experience to join a team. We'll try to set up teams of 3 to 6 members each. We're going to have 2 pitch meetings on July 13th and 25th. We'll ask people to upload a short presentation of 5 or 6 slides about the problem you want to model. We'll give you a slide deck to fill out for that. And also there'll be a document that you can fill out your information in. We want to go over the proposed projects to make sure that they're practical for CompuCell. CompuCell is quite powerful and flexible, but there's some things it doesn't do. It's not a molecular dynamics simulation. It's not a simulation of bones and biomechanics. It's not a simulation of electrical activity in the nervous system. And so we want to make sure that people's projects are feasible and so that people can make good progress in the hackathon and feel that they've accomplished something. Over the years, quite a few hackathon projects have turned into published research papers. And we want to make sure that we support that as much as possible. We'll make a preliminary list of projects that we think are a good idea. We won't prevent people from doing projects that we think are problematic, but we want to try to minimize frustration on everybody's part. We'll try to team people up. Again, people can reorganize. If people have late blooming ideas after the 25th, there'll be an opportunity at the workshop proper to pitch new ideas. But again, with these hackathon ideas, the sooner one can begin to organize an idea and a team, the more likely you are to make it work. During the hackathon itself, we'll have during the workshop brief noontime meetings on the hackathon ideas, teaming. During the hackathon itself, we'll have morning and noon check-ins, ask for brief written reports at the end of each day. At the end of the Sunday, we'll ask for oral presentations and model documentation from the hackathon teams. And we work closely with hackathon teams after the end of the workshop and hackathon to help people develop their ideas to something published. If you have hackathon ideas, please post them to the Slack channel. Please also add a description of your idea to the shared Google Docs. And when you're ready, you can upload your project pitch to the Google Drive, and we'll schedule it for presentation on the 13th and or the 25th and or during the workshop itself. The most important things now are to begin to think about what it is you want to model, what you want to learn from the model, why you want to do the model, and a little bit about your background and skill set, and maybe what you'd like to get out of your team members. We'll do our best to support you through the hackathon. Depending on how many teams there are, we may come up, some of the instructors may propose some hackathon ideas of their own. And again, if you're going to participate in the hackathon, you certainly don't need to have any biological, mathematical, or other knowledge, but it is important to have some basic coding under your belt before we get started. Okay, that was the main part of the logistics. I wanted to give a very quick fly-through of CompuCell. CompuCell consists of 2 primary tools: a smart model editor, which is built in with a lot of features that make coding very simple in CompuCell. In particular, almost everything that you need to do is pre-patterned in the editor, so you don't have to remember syntax of Python. And Player, which is a fairly flexible tool for simulating, steering, and visualizing simulations. I'll give you just a couple of words about installing and running CompuCell. CompuCell is cross-platform. It'll run on Windows, Linux, or Macs. However, especially in most recent Mac versions with the custom processors, it's a bit problematic. Some Linux installations are rather fussy. And so we're going to be teaching out of the Nanohub online version of CompuCell. If you have a strong difficulty getting CompuCell running locally, we would encourage you to use the online version, which doesn't require installation to your local computer. We can't promise, depending on how many we'll try, but we can't promise, depending on how many people show up, that we'll be able to help everybody troubleshoot installation issues on desktop and deployment. Again, if you're going to try to do it desktop, I encourage you to get it installed as early as possible, so we have more time to fix. Running CompuCell3D on Nanohub is pretty simple. And I'm going to switch to a split screen mode here, which is how most of the workshop will be taught, so that we can see both the slides and the program itself. Nanohub is a publicly funded tool in the United States. It's a server farm, rather old one at this point, funded initially by the National Science Foundation. And the great thing about Nanohub is it allows you to run programs from anywhere in the world for free. There are more modern online systems like Colab, but Nanohub has some unique features. It has a few annoyances, but we find it a very useful educational tool. And you can find the link to Nanohub, CompuCell3D.org/Nanohub. And at Nanohub itself, nanohub.org/tools CC3D base 4X. And we strongly encourage everybody as soon as possible to establish a Nanohub account. You go to the Nanohub homepage, nanohub.org, hit login or sign up, and it will take you to a set of screens. If you are in a US institution and you hit sign in with an affiliated institution, you should be able to create an account instantaneously. Otherwise, you can create an account through Google, and there are going to be some additional possibilities for account creation. You should be able to, wherever you are in the world, to create a Nanohub account. If you have problems with that, Nanohub has a help function. They're not always instant about getting back to you, but they're pretty good about coming back and helping you out. And so we strongly encourage people to do that. You could do that now if you haven't done it before. Once you've logged into Nanohub, you'll see something called the dashboard. It's a little bit primitive, but it looks like this. Here's my dashboard here on the right. I hope the screen share is working okay, so that people can see this. You can find your dashboard if you come up here to the top right and click on dashboard. And this is basically a navigating pane for controlling Nanohub projects. To launch CompuCell on Nanohub, the first time you're going to do it, you can go under All Tools, Search Tools. You can type CompuCell3D V4, and it'll pull up CompuCell's 3D V4 main tool. You can bookmark it by clicking this little heart box here, and you can launch it to run by clicking the file icon to the right. And so I'd ask people to do that, to follow along to do that. And what you'll see is that CompuCell will launch. It takes a minute for it to launch. Initially, you get a blue rectangle, and then it's going to pop up player, which will be the executable environment for CompuCell. If, on the other hand, you want to install CompuCell on your desktop, you can go to the CompuCell homepage here, download binaries. Here, you have Windows, Mac, and Linux releases. I'll go to Windows here. It says Windows binaries. I can click on the download button here, and that will in turn take me to SourceForge, where I can download a one-button installer for CompuCell. One thing to know for both Windows and Macs is that you will get a series of warnings when you try to do the CompuCell install, because we're not a registered Apple product or registered Windows product. And so you'll have to override the security settings to do the install. It is safe, but you need to do that. You may have to run us as an administrator to be able to do the install. And there are instructions, more detailed instructions on the website for doing the install in case you have problems. Again, if you're running some of the latest Macs, you may not be able to get CompuCell to run, in which case you need to do it online. I strongly recommend against compiling CompuCell. It's a major effort. Unless you're going to be becoming a power user developer of core CompuCell, you don't need to do compilation. One of the big things about CompuCell is you don't need to compile it. But if you really want to do it, the source code is available. It is an open source project. We love having super users who develop CompuCell core code. So if that's where you want to go with this, we would be delighted to talk. So again, here's the download. Again, also Windows 10 and Macs, you may need to run as an administrator. In the desktop version, you search for twit and open that as your gateway to CompuCell. Last thing I want to do today is do a quick exercise. I don't like to just lecture without doing something together. We're going to use twit to quickly create a simple simulation of growing cells, differentiating cells. And we'll do it inside of the Nanohub deployment of CompuCell. So let's come back to our Nanohub deployment of CompuCell here. And I'd like to do the poll, and I'll just make sure that everybody, or at least enough people, are able to get to the Nanohub CompuCell and get this running. And typically the way I'll work with this is I'll wait for a few minutes to see whether people are able to do the exercise. If you're not going to do it, please indicate that you're not doing it so I know. If you've done it, say done. If you need help, you've got an opportunity to ask for help. And if you need more time, please ask for more time. So somebody says they need help. If you could raise your hand. Then we could ask a question in the chat. And we can see if Joel or Hayden can help you out. I see people saying they need help, but I don't see any requests for help. So you have to help me out by knowing what you need. Okay, what can we do for you?
Yeah, just trying, I think I opened multiple links, so I just need to know the exact link. It's through Nanohub, right? The compute.
Yes, that's right.
Okay. Okay. So I think I'm a little bit, yeah, confused. Yeah.
So again, I'll put the link in the chat for you.
Yeah, thank you.
If we're not able to get it to work today, I wouldn't worry too much. You'll have plenty of time to go over it in practice beforehand, and then during the workshop, we'll go over this again. But absolutely, we don't want people to be frustrated or stuck. So one of the goals of this orientation is to try to make sure that people have the basics available so that they can learn effectively. Did that link work for you?
Yes, so it works. It's working now. Thank you.
Great. Okay. Okay. Are there any other problems or questions we want to address before we move on for a second? Okay. So you'll see this. This is a simulation editor, a simulation execution environment called Player. You'll find that you can move the screen. One problem you'll find in Nanohub sometimes is that the screen doesn't redraw properly. To get it to redraw, if you pull on the bottom right-hand corner of the bounding box, that forces a redraw and a screen update. The Compusell uses the X11 interface, which is a very, very, very old windowing system here on Nanohub. One thing that we will find is a problem is that you can't cut and paste text. You can cut and paste text within Compusell, but you can't cut and paste text from, for example, the chat function into Compusell. And this is unfortunately an intrinsic limitation in the Nanohub deployment. And we'll do our best to provide workarounds for that, but it is something that we a little bit of an annoyance. We're going to run the model editor now, and there are a couple of ways you could do that. This little pen and ink icon here, start Twit it, or we can go under a file menu and hit start Twit it, and you will see this open up. I mentioned resizing the window already, and you'll see a second window. You'll see, if you're on Nanohub, you'll see two tabs. One tab is the execution environment, one tab is the editor. And we're now going to create a new simulation. You can go under CC3D project, pull it down and select new CC3D project. And it'll pop up a dialog window where it'll ask you for the simulation name, simulation directory. And the first thing we have to do is type the simulation name. We can call it new simulation 3. Since this is the third time I'm doing this today, you could call it just new simulation. And we have to select the directory. If you're on a desktop machine, you have to select any writable directory. On Nanohub, you have to select your username here because you don't have write privileges to the root directory on their computer. So I'll select myself and hit next here. Oops, choose. And then I'll hit next. And this will now start defining a new simulation. The first window will be defining things like the lattice size, because CompuCell is a lattice simulation, and special properties. We'll leave this with its defaults. So we'll hit next. And it skipped. I'm sorry, my mouse is double clicking. It's got a bug in it. So if I skip something, it's because I've got a mouse that a single mouse click registers multiple clicks. The next thing we'll do is list the cell types we're going to simulate. In a real simulation, we'd give these names like cancer cell, epithelial cell, immune cell. Here we can just call them cell type 1, cell 1, hit add. Cell 2, we're going to create two cell types, cell 1 and 2, hit add. So please follow along with that. We hit next. We're not going to use any chemical fields in the simulation, so we'll skip that. And then we're going to specify the properties we want our cells to have. We're going to have them stick to each other, that's contact adhesion. We're going to have them grow, growth. We're going to have them divide, mitosis. And because in these simulations, nothing exists unless we assert it, we're going to say that they have a volume. So we need to click contact, growth, mitosis, and volume as tick boxes. And I'll come back and I'll redo the poll, see if people are able to keep up with that. And again, if people are having trouble or need help, please speak up, but it looks like people are able to keep up. We'll hit next. It'll tell us the configuration is complete, and we hit finish, and you'll see a screen like that. The simulation is here. To view the simulation, I double-click on the simulation name in the left-hand navigation panel, and it'll open up a series of windows that contain the simulation specification. I can use this double floppy disk icon to save the simulation, or I can go under CC3D project. I can name the simulation something else if I want to version. If I want to run the simulation, I have a number of options. I can go over the simulation name here, right-click, and say Open in Player. I can also go under CC3D Project and select Open in Player. Either one will work. I'm going to do, since I've got it open, I'll do Open in Player here. And then in Nanohub, on desktop, it will open the player separately. In Nanohub, I have to switch back to my other tab. Here's my simulation tab. And I should be able to view the simulation. It should look something like this. And you can see the simulation is actually executing. It's at step 400 already here. Now, I'm going to stop that simulation and restart it so that we can see how it looks at the beginning. You'll notice that the drawing is, the size of the box isn't the most convenient. I can use the resize functions here to change the size of my box. I can use the maximize here. I can move the bounding boxes easily enough to rearrange things the way I like. One tip that's very useful is if I go under the Windows menu and I select Tile here, it will relay out the screen given all the menus and boxes that are available, and it'll try to optimize the layout. And so if you have a bit of a mess on your screen, that tile function comes in handy. When I'm going to control the simulation, I have 4 buttons here. The left-hand button, they're based on cassette players, so rather old-fashioned icons. The left-hand button plays the simulation, runs the simulation. The button here with the 2 vertical stripes is a pause button that halts the simulation temporarily, allows you to restart it, to make changes to it or to look at it in detail. The thing that looks like a record button actually single steps the simulation. It does 1 time step of the simulation. The stop button I recommend not using unless you have to, because it resets the simulation to the beginning. So you lose all the information in the simulation. Sometimes you need to stop and start again. But in general, while you're playing with the simulation, you don't want to use the stop function. And so you've now written the simulation. It's created an array of 2 cell types, 1 and 2. And if I now hit run, I will see those cells grow. And if I zoom in, I will see I have 2 cell types, and let's do some steps. I see the cells growing, and I'll see the cells divide. And if I look carefully, I'll notice that when cells divide, one of the cells, that's a daughter cell, stays the same color as the parent, and the other one will change color to the opposite color. And so the last thing I'd like to do would be to come back and tell you how that little simulation works. Compusell simulations have 2 main parts, a CC3D ML markup language part for static properties of the simulation, and a Python part that does the dynamics. That's why we need to learn a little bit of Python. Within the simulation environment, once I've opened the simulation here, I have 3 tabs, XML tab, and something called new simulation steppables.py, which is the main Python tab. There's also something called new simulation.py, which typically we won't look at. We'll learn about that later in the course, but most of the time that's handled for you automatically. I can also navigating by double-clicking in the navigation tab here on the left. You can use the scroll bar here on the right to move through the code. Let's look at the XML. The first few lines of the XML have metadata about parallelization because Compusell is intrinsically multi-threaded, and the lattice information. How big is my lattice? How long does the simulation run for by default? And some other lattice specifications. For example, here it says that the lattice is 256 by 256 by 1. You'll see my array is 256 by 256. The second block of code tells me that I have a cell type medium. You're always going to have a cell type medium. That's what's present when you don't define cells. Think of it as the fluid around the tissue. And then two cell types, cell type 1 and cell type 2 that I've defined. And then a statement that the cells have a volume because nothing exists unless I do it. These are called plugins, and plugins define the intrinsic properties of the objects in the simulation. Those are going to be the things that define how cells behave and interact. If I look in more detail, you'll see that if I go back to the player, there's a map that says my cell types and the colors associated with them on the display. The next thing that I have in my specification is contact energies. This tells me how much the various components, cell, medium, cell type 1 and cell type 2 stick to each other. The default is they all stick in the same way, which is not very exciting, but I can go in and I could edit this in the executable, for example, by changing the contact energies as the simulation runs. So for example, if I wanted to change the energy between cell type 1 and cell type 1, I could type in a new value here. So this means I can change parameter values in the simulation as the simulation is executing. That's relatively unusual in these kinds of simulations. Typically, once you've specified the simulation, it's static. Compusal allows you to do what's called steering, that is changing the behaviors of the simulation as it's executing. Yep. The next thing we'll see is something called a steppable. These are things that are executed in alternation with the way the cells are moved, typically implemented in Python. This is called uniform initializer. Compusell doesn't have tremendously sophisticated internals for defining cell conditions, but this will do a layout of cells. It will draw a square box or rectangular box. I can specify the size of the cells here, specify the size of the cells, whether there's a space between them and what kinds of cells I want to have in my simulation. And this specified that square grid of two cell types that I started my simulation. The second part of the simulation is the Python, and I'll give you just a little bit of guidance about what the Python looks like. Again, if this is unfamiliar, we'll teach a Python module to get you up to speed. The Python will consist of two main functions, a start function, which is run once at the beginning of the simulation that initializes conditions. Here I have an iterator for cell and self.cell list that does something to every cell in the simulation. And here I'm assigning the cells something called the target volume. You can think about that effectively as the mass of the cell, and the cell lambda volume, which is the compressibility, or actually 1 over the compressibility of the cell. The next thing I do is I have a step function. The step function is the core of the simulation. And my step function here goes through my cell list again, and it increments the target volume of each cell. That means the cells are all growing at a constant rate. You could change that and see what happens if we had a little more time. The last thing that we do is we want our cells to divide. It's a little bit more complicated. Here we create an empty list. This is a Python concept. Square brackets are lists of cells to divide. We go through our list of cells. We ask if the cell volume has reached a threshold. In reality, cell division is more complicated than that, but it's not a bad place to start. If we've reached that volume threshold, we add our cell to our list of cells to divide. Then we go through the cells that are dividing. We have a variety of functions that allow us to make the cells divide with random orientation or in various fixed planes or along major or minor axes of the cells. Once the cell has divided, we have to say what happens because we've created a new cell. Typically, we have both the parent and the daughter cell take half of the volume of the original cell. And we clone the attributes. That is, the daughter cell has the same attributes as the parent. In this case, we then go ahead and we say if the parent cell was of type 1, we make the daughter cell of type 2. The parent cell was of type 2, we make the daughter cell of type 1. If you run the simulation and watch, you'll see the parent cell keeps its identity as cell type 1 or 2. The daughter cell will swap colors with the parent. And that's the entire simulation. So that is building and running a simulation in a couple of minutes. A little bit of a flavor that you can explore more. Preparing for the class, please make sure that you can launch CC3D either locally or on Nanohub. You can play with editing, loading, and running simulations in Twit, like the one we just did. Compusell comes packaged with a variety of demonstrations. There's a nice one on steppable-based mitosis. It's a little bit of an extension of what I did here. There's a nice simulation of angiogenesis that you can play with, and a simulation of delta notch signaling, which will explore how to use networks and signaling models. We encourage you to do a little bit of background reading in the areas that we've talked about that you may not feel comfortable with. And if you're planning and participating in the hackathon, please do start working up a project you might want to do, or think about the project you might want to join as a team member. And that's all I have for today. I appreciate people sticking around. If there are any questions or things about the hackathon you'd like to talk about now, feel free to unmute and ask away.
I do have a question. So, like when you just went through like the demo and the code is essentially like generated, are we going to be able, that allows us to like customize things further and add parameters potentially, or would that, if we add too many things at this level, would that like mess up, I guess, the basis how this was formed? Or will we just have to go back to that screen where we checked off of those certain conditions? Do we need to do most of it there, or will we be interfacing with both, like a checklist and actual coding?
You'll be doing both. I wish we had a full GUI model specification environment. We're working on that. But at the moment, what we do is Wizard will give you a template, which will give you the basic structure of your simulation. Then what you'll have to do is you'll go in and you'll edit with the inside of the steppables. You'll be editing. And what you'll find is that Twit has some nice features, which is that if I go to Twit and go to CC3D Python, there's a long list. And the hardest part here is remembering where things are because there are too many menus. It's a little bit like Microsoft menus. Suppose that I want to visit every cell of a given type. So I want to do something not to every cell, but to all cells that are cell type 1. I can paste in the code here, and it'll give me a boilerplate code for doing that. Suppose that I wanted to add go back to my starting function here, my start function. Suppose that I wanted my cells in my start function to have a a subcellular model. I can add an antibody model to my cells, and this will give me the basic code to add a network model to my cells. And so you still have to do some programming, but many of the most common functions, suppose that I wanted to create a scientific plot of a parameter in my simulation, a variable in my simulation. The code to do that is present. I'll have to edit it to say which variable I want to plot, how frequently I want to plot. But the basic templates for almost everything that you want to do are present. And so you'll be doing coding, but there's a lot of assistance in the editor to make the coding easier. So we can't prevent mistakes. And of course, you have to think about what it is you want to model. But the editor is designed to make the actual process of coding as easy as possible. There also are similar help for doing the markup language changes. And also, if you actually were writing hard-coded C++, there are templates for that as well. So this editor, I think, is a big plus of CompuCell. It means that you have the advantage of being able to do free coding without having to remember everything. Helps avoid a lot of mistakes. Any other questions, comments before we finish up? Okay, well, I want to thank you for sticking it out. Appreciate it. I know, depending on what time zone you're in, it may be early, late, or sometime in between. I look forward to seeing those of you who are interested in Hackathon on the 13th and the 25th. And I'll see the rest of you on the 31st or the 1st. And I do appreciate everybody joining us and being willing to talk. And I hope that this will be an interesting experience for all of you. Thanks very much.